All right. Hello, everyone. Welcome to the last lecture of the class on the reproductive system. I'm going to go ahead and share my screen, and we're going to go ahead and talk a little bit about the reproductive system. All right. So the reproductive system is, like most of the organ systems in this class, fairly complicated, and we could go into quite a bit of detail. You know, much like some of the other organ systems that we've talked about so far over the last couple of weeks, we could really devote an entire semester to this. So what we're going to be doing in this lecture is we're going to be looking at some of the high points, if you will. We're going to be talking about the anatomy of the male and the female reproductive systems. We're going to be talking a little bit about how gametes are made, specifically how sperm and ova or eggs are made, spermatogenesis and oogenesis. We're going to focus a little bit more on the female reproductive system because we're going to be talking about the uterine and the ovarian cycles, and then we'll very briefly mention procreation and childbirth, although those are a little bit outside of the scope of this class, so we're not going to spend a lot of time on those. All right. So let's go ahead and dive into the anatomy of the reproductive system. With both the male and the female reproductive systems, we're looking at a situation where we are focused on moving the reproductive cells, either the sperm or the eggs, these are the gametes, out of the body. So in both of our reproductive systems, that's going to be the goal. All right. We see that even though there's a little bit of shared embryological origin with some male and female structures, and I'll talk about that in just a couple of moments, these two anatomies are really quite different from each other. So this is the one organ system, unfortunately, where you're really going to be forced to learn two separate anatomies instead of just one, because all of the others are the same in both males and females. So we're going to go ahead and start with the male reproductive system that we see right here. And we're going to go ahead and start with the gonad, the testes. So I'm going to pull up my pins here, and we're going to go ahead and start here with the testes. We'll take a little bit of a closer look in just a couple of moments inside of the testes. But suffice it to say for right now, this is where sperm are being made. The testes are housed outside of the body cavity in a sac of skin called the scrotum. The reason behind, or the reasoning behind the evolution of a scrotum is that sperm can't be made at body temperature. They have to be produced at a little bit lower than body temperature. So the testes are housed outside of the body because sperm cannot be made at body temperature. Now, most mammals have the ability to internalize their testes. This is kind of corresponds with mating season. When mating season comes around, the testes descend into the scrotum, and during the non-mating season, the testes will actually ascend back into the body cavity. And of course, the reason behind this is to protect the testes. They're arguably a fairly delicate organ, and the scrotum really doesn't offer any protection at all. So with that in mind, it's really kind of the utmost importance to protect those and not have them out year-round. Now, primates and humans especially don't really have a defined mating season. And as a result of that, they are going to have the testes out on a more or less continuous basis. So we don't really have them ascending into the body cavity or descending into the scrotum. They stay out pretty much all year. Now, of course, this does run a little bit of a risk, for damage to this organ, but thankfully the injury rate is not terribly high. As sperm are produced, and again, I'll talk about spermatogenesis a little bit more later on in this lecture. But as sperm are produced, they travel out through a series of tubes. We're going to look at the internal anatomy of the testes in just a moment. We're going to see that it really is just miles and miles of these little seminiferous tubules. They eventually are going to converge in the structure that we see right here called the epididymis, which is this structure that we see right here. And then the epididymis sort of turns into this structure that leads us out of the scrotum. This is called the vas deferens. It's also sometimes called the ductus deferens. And then the vas deferens are going to carry the sperm back into the body cavity. Once they've been produced, they can be raised to body temperature. It's just the actual production of the sperm that needs to be just a little bit cooler. The vas deferens is the primary site of male sterilization, right? If a male or a man decides that he doesn't want to have kids or wants to take an active role in family planning, he might opt to get a vasectomy, right? Where the vas deferens is cut. Typically what they'll do is they will make a small incision in the scrotum and they will cut the vas deferens. And that way it blocks the exit route for the sperm. Okay, we're going to talk about semen in just a couple of minutes. The vasectomy doesn't really affect the production of semen or any sexual performance, but it does block that exit of the sperm out of the body, and that's intended to be a more or less permanent form of contraception. Although in certain circumstances, vasectomies can be reversed, where they would just connect those two ends of the vas deferens again, and then you would have potentially the ability to have children again. So the vas deferens runs up into the abdominal pelvic cavity. And kind of right below the urinary bladder, right in this region right here, we see that the sperm are going to be mixed with the secretions of a couple of different organs. There are paired seminal vesicles that we see right here. Okay, there are paired seminal vesicles. And then there's the prostate gland right here. All right. These two structures provide secretions that account for almost all of semen production. All right, so the seminal vesicles and the prostate gland each contribute about half of the volume of semen, with the seminal vesicles being a little bit more. We'll talk about that in just a little bit as well. And then at that point, the semen with the sperm can leave the body through the urethra and eventually exiting out the end of the penis. The end of the penis is called the glans penis. You'll notice here there's this prepuce of the penis. The prepuce is what we kind of colloquially call the foreskin of the penis, and it's what's removed when when we circumcise a baby. It's that prepuce that's being cut off. The idea behind circumcision is that the space here between the glands and the foreskin could potentially harbor bacteria and things like that, leading to increased risk of STDs. And in some countries, that's been shown to be the case. Not in every country, though. All right. So at any rate, we have this kind of glands. We have this plumbing system out of the body. You will notice that here in the prostate gland, right in here in the prostate gland, we start to share plumbing with the urinary system. The urinary bladder is this structure right here, and urine exits the body out the urethra as well. So there is a little bit of shared plumbing here between the urinary system and the reproductive system. As a result of that, because sperm are still fairly fragile, even though they can be raised to body temperature, they still really react to changes in their environment, and not always the best way. What we do is we have to cleanse the urethra before ejaculation occurs. And the cleansing secretion is created by this really small gland back here called the bulbourethral gland. It's also sometimes called the Cowper's gland. But this bulbourethral gland produces a secretion that's actually passes through the penis before ejaculation, and that allows to get rid of any residual urine and stuff like that that could potentially cause a problem with sperm viability. So several glands are at play here. The bulbourethral gland produces that fluid that comes out before ejaculation. And then we have the seminal vesicles and the prostate gland, both contributing to the semen volume that we see here. I'm going to erase a couple of my markings just a little bit here. One thing you will notice here is that the prostate gland is cut in half right here, so we can kind of see inside of it. But what it does is it actually wraps around the urethra, right? So if the urethra was right here, the prostate gland would kind of wrap around it like this. So one of the things that is fairly common, in fact, I think the statistics say that it affects something like 1 in 3 men, is an enlarged prostate gland. Okay. If your prostate gland becomes swollen or enlarged, what that will do is it will actually pinch the urethra, and that can cause all sorts of problems with urination. It can be difficult to start urinating. It can also be difficult to control when you urinate, and that can be really problematic. And that enlarged prostate, for whatever reason, can affect almost a majority of men at some point in their lives. So it's important to sort of keep up with that. Of course, the penis itself is the organ that's going to deliver semen to the vagina. It's made up largely of erectile tissue. There are several chambers in the penis that are going to fill with blood during an erection and allow it to be inserted into the vagina. Let's take a little closer look inside of the testes. So as I mentioned, the testes are really just a big conglomeration of tubules. There are these seminiferous tubules in the testes that are responsible for the production of sperm. And there literally are miles of these tubules in each of the 2 testes. And then surrounding the testes, of course, is a really dense connective tissue called the tunica albuginea. When the sperm are made, as I mentioned, those tubules more or less coalesce into this structure called the epididymis, and then the epididymis is going to become the vas deferens. The vas deferens is packaged into a larger structure called the spermatic cord that also has blood vessels packaged with it. The testes both have a really rich blood supply. And in fact, the way that the blood works, it actually works as a little bit of an air conditioner for the testes. And we keep kind of cooler blood down near the testes, and then we warm it back up as it comes into the body cavity. So that also helps to ensure that the testes are kept just a little bit cooler than body temperature. In the connective tissue that makes the testes up, it's all this white area that you sort of see here between the tubules. There are additional cells that are responsible for the production of testosterone. Those are called interstitial cells, and they lie between those seminiferous tubules. So in addition to making sperm, one of the things that we associate our testes with is the production of testosterone. Right. Now, testosterone is necessary for the production of sperm, and we don't really start making large amounts of testosterone until puberty. If you remember back to my endocrine system lecture, I mentioned that the adrenal glands make very small amounts of testosterone throughout life in both genders. But in males, we see that once puberty begins, that's when the testes really amp up their production of testosterone. So again, it's responsible for sperm production. However, it also is responsible for the enhancement, I guess, of secondary sex characteristics. So in biology, when we talk about secondary sexual characteristics, we can contrast those with primary sexual characteristics. Okay. Our primary sex characteristics are our external genitalia. So in males, we're looking at the penis and the scrotum. In females, primary sexual characteristics are the clitoris and the labia. Secondary sexual characteristics are anything that we associate with maleness or femaleness that is not part of the genitalia. So in males, we can talk about things like the deepening of the voice, increased hair growth. Now, that kind of applies to both genders, but it also talks about, we also can talk about localization of hair growth, right? Things like facial hair growth is specific to men, whereas axillary, armpit hair, pubic hair, those are going to be both males and females. In addition to that, we also see that males become more muscular when they hit puberty. We see the enlargement of skeletal muscles, and we also see an increased heaviness of the skeleton as we start to go through this. And the primary purpose of that, of course, is to support the increased skeletal muscle mass, right? So, I sometimes joke that if you're ever confused about a secondary sexual characteristic, find something that gets awkward during puberty, and that's probably a secondary sexual characteristic, right? The deepening of the voice, as it deepens, it starts to crack. That can be very awkward. The increased hair growth. Before you can grow a full beard, you have to kind of go through that period where you look like Wolfman Jack and stuff like that. So, at any rate, that's the joke that I always sort of make. We'll talk about female secondary sexual characteristics here in a couple of minutes. As far as those accessory glands go, like I said, and I've already explained this, I'm not going to spend a lot of time on this slide. We have the seminal vesicles that are responsible for producing the majority of semen volume, about 60%. The secretions that the seminal vesicle has are really rich in sugar and vitamin C. Vitamin C plays an important role in helping to buffer the sperm against the acidic environment inside of the vagina. Prostate gland has that milky fluid that plays a role in activating sperm. And then, as I mentioned, those bulbourethral glands produce this clear fluid passing down the urethra before ejaculation. So we'll come back to some of this stuff in just a couple of minutes. Let's go ahead first and take a look at the female anatomy, which is, as I mentioned, quite a bit different than the male anatomy. The first thing that is perhaps the most striking is that there's no shared plumbing between the female reproductive tract and the urinary system. These are completely separate in the female anatomy. The other thing that I think is really noticeable is that the gonads, the ovaries, are not externalized. They're completely internalized in females. Eggs or ova can be produced with no problems at all at body temperature. So there's no reason to externalize them. So we do see two gonads. We see two ovaries that are in the abdominal pelvic cavity. Those ovaries are suspended within the cavity by a series of ligaments, and they're not directly connected to the duct system that we see. Okay, now the testes were directly connected via the epididymis and the vas deferens. Okay, those seminiferous tubules had a direct connection. So there's kind of a closed plumbing line there. The ovaries are a little bit different, where they're not perfectly connected. They're very close proximity to the duct system, but they're not completely connected. So, of course, the ovaries are where ova are made. We're going to talk about the ovarian cycle a little bit later on in this lecture. But suffice it to say for right now that when an ovum is mature, it becomes ovulated, okay, or it ovulates from the ovary. It's ejected from the ovary. At this point, it could come into contact with a sperm. So what happens is that it needs to find its way into this duct system that we see right here. Right? These ducts, of course, are connected to the uterus, which is right here. This is where pregnancy will occur. the uterus is separated from the vagina by this opening called the cervix. And then of course you have the vagina right here, which will receive the penis during intercourse. But the other end of the tube, the other end of these fallopian or uterine tubes, are not perfectly connected to the ovary. They surround the ovary, but there's not a direct connection there, right? So when an egg is ovulated, more often than not, it gets trapped by the opening of this tube. There are these finger-like projections called the fimbriae that you see right here. What they do is they kind of try to pull an egg in towards them. And 99.9% of the time, they end up getting swept into the fallopian tube and ultimately into the uterus. Okay. We'll talk about it in a little bit. More often than not, fertilization occurs approximately here in the fallopian tube. But eventually the embryo would make it down to the uterus. Okay. If the egg is not trapped by the fimbriae, it will float out into the abdominal pelvic cavity. And again, more often than not, nothing really happens. However, there are some instances where an egg that kind of floats out into the abdominal pelvic cavity can latch on to an internal organ. Even a fertilized egg could end up in the abdominal pelvic cavity, and it could start to grow on something else. That's called an ectopic pregnancy and can actually be life-threatening for the mom. So we need to address that as quickly as possible. But like I said, 99 times out of 100, 999 times out of 1,000, things go as they should. But I just want you to be aware that there's not a direct connection. The uterus, like the ovaries, is suspended within the abdominal pelvic cavity by a fairly complex series of ligaments that you see right here. Okay. Undo. There we go. Okay, so the ovaries are the site of ovum development. I'll come back to this slide in just a little bit of time, okay? They also produce estrogen, which is responsible for female secondary sexual characteristics. I'm gonna skip ahead just a little bit. Female secondary sexual characteristics include the development of breasts, the appearance of axillary and pubic hair, increased deposits of fat around the hips and the breast, widening and lightening of the pelvis, and of course, the onset of menses. Right. Again, we'll talk about that in just a little bit as well. All right. So that's what our anatomy looks like. I mentioned that even though these are different, there is sort of some embryologically shared structures. So really early in our development, very early in our embryonic development, like within weeks of fertilization, we end up with what we call a bipotential gonad. And the word bipotential just means it could become one of two things. Okay, and as you can probably guess, it could become either an ovary or a testis. All right? So we have this bipotential gonad. We also have a few other things that I'll talk about in just a couple of minutes, but we have two sets of ducts. And the names of those ducts don't really matter, but we have two sets of ducts as well. All right? So the idea is that if you are genetically male, and I'll talk about our genetic makeup here in just a couple of minutes, but probably all are aware that males have an X and a Y chromosome, and females have two X chromosomes. These are the sex chromosomes. If you are XY, if you are genetically male, that Y chromosome has genes on it that are going to guide the development of that bipotential gonad into a testis. Okay. If that gene is absent, in other words, if there's not a Y chromosome present, the default of that bipotential gonad is to become an ovary. Okay. The Y chromosome will also guide the development of that one set of ducts that will become the epididymis and the vas deferens. If the Y chromosome is absent, the set of ducts that would become the fallopian tubes and the upper part of the uterus is what's going to actually develop. Okay. So there's one gonad, it's either going to become an ovary or a testis, and then there are two sets of ducts, and either the male set or the female set is going to develop. As far as the external genitalia goes, there's significantly more shared structures. So as far as the external genitalia goes, we have these folds of skin. They'll either become the scrotum or they'll become the labia, depending again on the Y chromosome. And then we also have this mass of erectile tissue as well. If the Y chromosome is present, that erectile tissue will become the penis. If it's absent, that erectile tissue will become the clitoris, which, like the penis, has the ability to become engorged with blood and become erect during sexual stimulation. All right. Okay, so definitely some interesting shared structures that we see here. As far as the human life cycle goes, I want to kind of go through some vocabulary here, and then we can talk about spermatogenesis and oogenesis. So here we are these adults, right? These people that we have here. And we know that we have two genders. We have a female and a male. Okay. Regardless of that, we are in this phase of our life cycle called the diploid phase. What does it mean to be in the diploid phase? Well, all we're referring to there is how many sets of DNA you have. Okay, and you're probably aware that you have half of your mom's DNA, you have half of your dad's DNA. Right? So, long story short, is you have two sets of DNA. Right? You have a set from mom and you have a set from dad. Now, the way that those two sets interact to make you is the subject of a genetics class. But when I talk about having two sets of DNA, the term that I give to that is that I am diploid. Again, that prefix di means two. Okay. We can denote it in other ways as well. The other way that we denote diploid is shorthand, just saying that you are 2N. Okay. Our diploid number or our ploidy number is 46. Okay, that means that you have a total of 46 chromosomes. Okay, now remember, you get half of them from mom, you get half from dad. So you have 23 from mom and you have 23 from dad. Of those 23, one of them that dad gave you is a sex chromosome, one of them that mom gave you is a sex chromosome. And of course, that's what determines your biological gender. Okay, so that's, or your biological sex, I guess I should say. All right. So essentially, that's what we have here. Is we have these 46 chromosomes. Of those 46 chromosomes, 2 of them are sex chromosomes. The remaining 44 are what we call autosomes. Okay. In order to reproduce, we have to produce cells that have half of our DNA. So we have to produce gametes, okay? Females produce eggs, males produce sperm. Okay? In order to get from a diploid cell to what we call a haploid cell, we have to undergo a process called meiosis. Now, I'm not going to make you learn all of the steps of meiosis in this class. I think that Biology 1 or a genetics class would be maybe a better place to learn that. But suffice it to say, you're making a cell that has half of your DNA, okay? When you make that sperm or that egg that has half of your DNA, you're not just passing on your mom's DNA or your dad's DNA. You're passing on a combination of their DNA. And, as you can probably guess, not all of your sperm are going to have the same DNA in them. Not all of your eggs are going to have the same DNA in them. Our biology has been very good to us in the sense that we can mix all of that up in infinite ways in order to increase the diversity of the offspring that we produce. And The reasoning for that is the subject of an evolution class. But again, that's essentially what we're doing here. So what we end up with is these sperm and these eggs, these gametes that have 22 autosomes and a sex chromosome. So for a total of 23. Okay. You'll notice that the shorthand for this haploid cell is simply N. Okay. It's worth noting that not all species have the same ploidy number. Some species have a few more chromosomes, some have a few less, but humans have 23. When the sperm and the egg combine in fertilization, we end up with a diploid zygote that now has 46 total chromosomes that can undergo mitosis and develop into another individual. So we spend most of our life cycle in our diploid phase, very short amount of time in the haploid phase. Right? But it's important to understand how these are made. What I'm going to do is I'm going to give you guys a brief tour of both spermatogenesis and oogenesis. The details we're going to really simplify, because this is the time where we could spend just a ton of time talking about this. Okay, so we're really going to simplify the details here, and there are a few takeaways that I want you to really pay attention to. So I'm going to go through some of the high points of both of these, and then what we'll do is we'll talk about some of the main points of comparison and contrast. So let's go ahead and talk first about spermatogenesis. Spermatogenesis, the production of sperm, as I said, takes place in the seminiferous tubules that make up the majority of the testes. If we take a cross section of one of those tubules, like we've done right here, we start here at the basement membrane of that tubule, and we start to work our way towards the lumen. This is the lumen of the seminiferous tubule right here, where these sperm that you can see that are being made are eventually going to work their way to the epididymis, work their way to the vas deferens, and eventually out of the body. All right. One thing that you'll notice is that the process begins with two end cells, diploid cells here. These stem cells, or in the case of spermatogenesis, spermatogonia, are just like any of your other cells. They have two sets of DNA in them. So they're just the same as a skin cell, a brain cell, a muscle cell, whatever, right? They have two sets of DNA, half from mom, half from dad. The process of spermatogenesis begins when that spermatogonium undergoes mitosis, okay? So you produce two identical daughter cells. One of those daughter cells is going to stay at the basement membrane and continue to undergo mitosis. The other one is going to enter meiosis. All right, and that's an important point. We're going to come back to that in just a couple of minutes. One of those daughter cells is going to enter meiosis. The other one is going to stay in the basement membrane. So I'll come back to that in just a couple of minutes. Okay, that one that enters meiosis is going to start to undergo its meiotic divisions, and as it does this, you'll notice that it is starting to look more and more sperm-like. Okay. Again, all the individual phases of meiosis are things that we're going to discuss a little bit later on. Okay. One thing worth noting right now is those seminiferous tubules. It's not a fast process. The process of sperm production takes place 74 to 72 days. I think I have a typo on the slide. I think 64 to 72 is probably the takeaway here. We can simplify that by saying it takes about 2 months for this spermatogonia, this daughter cell, to actually become sperm that are ejected into the lumen of the seminiferous tubule. You'll notice that by definition, from the process of meiosis, that single diploid cell will produce 1, 2, 3, 4 sperm cells. Okay, this process does not begin until puberty. So nothing here is happening until puberty. But again, once in the process of testosterone and follicle stimulating hormone from the pituitary gland, this process will commence. Okay. Once it begins, though, it is essentially going to be happening nonstop. Okay, it takes 2 to 2.5 months for us to make the actual sperm, but it's happening continuously once puberty begins. Okay, remember, we had that one daughter cell that hung out in the basement membrane. That one daughter cell is going to continue to undergo mitosis. So in theory, from puberty until death, men can make sperm. This is a nonstop, continuous process from puberty until death. Now, we can see natural testosterone loss over time and stuff like that. Sperm production can fall off as we age, but the process, in theory, will continue indefinitely. All right. So even though we see that the process itself takes a significant amount of time, it's happening continuously. So men make, on average, about 200 million sperm every single day. I want you to wrap your mind around that number for a minute. 200 million sperm every single day. That's utterly remarkable. Okay. Now, when they are ejected into the seminiferous tubules, they look like sperm. These look pretty sperm-like here, but they aren't able to actually fertilize an egg at this point. They're what we call incapacitated at this point. It's not until we actually mix them with the secretions of the prostate gland that they become capacitated and can actually fertilize an egg. That's kind of a long way of saying that a sperm has to be ejaculated in order to be able to fertilize an egg. The sperm themselves have a fairly simple anatomy. They have 3 primary parts to them. They have what we call a head, they have a midpiece, and then they have a tail. Okay. The head, of course, is going to contain the nucleus. That's the genetic material. There's also a cap on the head of the sperm called the acrosome. The acrosome contains enzymes that will allow that sperm to penetrate the egg when it reaches it. The midpiece contains mitochondria. It's a long swim for this little cell from the point of ejaculation to the egg, which is in the fallopian tube. And as a result of that, it's going to need a lot of energy. So we're very rich in mitochondria. And then, of course, the tail is the flagellum. Okay. All right. Oogenesis is a little bit different. I'm actually going to do this really quickly. I'm going to let you see just about everything. Okay. Remember, spermatogenesis didn't begin until puberty. Nothing happened. Oogenesis, the production of eggs, actually begins before birth. Okay. So what we see is a very sort of stop-gap sort of a situation here. We see that the oogenesis begins before birth. At birth, the process stops. It comes to a complete halt. And then once puberty begins, the process begins again. Okay, so we see about a 12 to 13-year-old or 12 to 13 long gap in this production of ova. All right. The ova, the eggs themselves, develop within a structure called follicles. So in addition to looking at the production of the ova, we're also going to be looking at the development of this structure called a follicle. All right. So bear with me as we kind of go through this. And again, we're going to be looking at some of the at the high points here. So again, we have these stem cells called oogonia in this case. All right. We have these oogonia here. And before birth, these oogonia are going to enter meiosis. Okay, they're still diploid cells at this point, but they have entered the very first phase of meiosis called prophase 1. If you remember from spermatogenesis, the spermatogonia underwent mitosis before they entered meiosis. Here, every single one of these enters meiosis without a mitotic division. So what this means is that all of those oogonia are going to just from the very beginning start their path towards becoming an egg or an ovum. All right? This is really key. You might have heard at some point in your life that women are born with all of the eggs that they're ever going to have. And that's absolutely true. because there's not a mitotic division that helps to keep these stem cells kind of stocked up in the ovaries. All of them are going to enter meiosis before birth. Okay. Now, a woman is still born with way more ova than she'll ever need. Okay. I think the rough estimate is something like 20,000 per ovary or something like that. And if you do the math, if a woman is having ovulations from age 12 to 60, that's one per month for almost 50 years. We don't even come close to reaching that mark of 20,000. But it's definitely something worth considering here, is that there's a big primary difference that we see right here. Okay. Once these oocytes, though, are reach this phase of prophase 1, that's what happens at birth, and then nothing happens for 12 to 13 years until the onset of menses. Okay. There is a small follicle that's developing around that ovum, and they're called primary follicles, very, very small little structures. Okay. Once we reach puberty, and once we see the onset of menses, what we're going to see is, again, a fairly lockstep sort of a process. Remember, you've got 20,000 of these primary oocytes that are in the ovary, ready to go. Okay, but once puberty begins, we're going to see one of them, on average, becoming mature per month. Okay, not all 20,000 of them at once, one. Okay, granted, there are instances where you can see more than one. That's where we see things like twins and triplets and so on and so forth kind of coming about. But on average, we're looking at one per month. Okay, and then what we see here is, from puberty to menopause, we see that we end up with the completion of meiosis. Okay. Now, with the sperm, and in the way that we normally teach meiosis, we tell you that there's going to be 4 haploid cells that are produced. Here, we only see the production of a single ovum. Okay. The rest of what would be ovum are actually going to degenerate as things called polar bodies, and they're not actually going to become eggs. So they're never going to quite develop as much. Okay. It's also worth noting that last meiotic division doesn't complete until the egg is actually fertilized by a sperm. So there's a lot of what ifs that have to occur in order for an ovum to actually be viable. If there's no sperm in the system, the egg is actually never viable. Okay, it has to be fertilized by a sperm in order to complete its second meiotic division. Okay. You'll notice here on the right-hand side of the screen that as we move through those meiotic divisions, the follicle gets bigger and bigger and bigger. Its biggest size that we see right here is where we see it being called a mature or Graafian follicle. And then after it ovulates, that follicle will actually disintegrate into something that we call a corpus luteum, and eventually a corpus albicans. And that corpus luteum is going to be really important because it's going to secrete a hormone called progesterone, which is going to help us to maintain pregnancy. All right. So what are some of the differences that we see here? Well, I want you to think about the comparison and the contrast of these two processes, right? When does spermatogenesis begin versus when does oogenesis begin? Right? Spermatogenesis begins at puberty. Oogenesis technically begins before birth. How many cells are made? Right? The spermatogenesis, we see 4 sperm from every stem cell. With oogenesis, we only see that single sperm, or that single egg. I'm sorry. Okay. And then, there's a couple of other things as well, but those are kind of the important ones. Okay. Here we see that fertilization has to occur in order for the egg to become viable. Not so with sperm; they're viable from ejaculation onward. All right. So definitely some interesting sort of parallels, but I think the differences are also kind of interesting. One thing worth noting is that women essentially get 1 chance per month on average to reproduce. Men produce 200 million sperm a day. That's 200 million opportunities to reproduce every single month. There's a really big disparity there. And as a result of that, and that isn't just true in humans, it's true in most species as well, where the women or the female is sort of the limited gender, and the male is sort of the one that has unlimited opportunities to reproduce. And that creates all sorts of interesting social situations and things like that, because females having only one chance to reproduce are typically going to be the choosier of the genders, whereas the males are going to be more competitive. And that creates all sorts of interesting social situations and things like that. That's why we often see, like with birds, the males are very flashy and stuff like that, because they're competing for access to females. That's why we see big bloody battles and things like elephant seals and things like that as well. So a little bit more about the female reproductive system, okay? Because we have kind of a lot happening here in the ovaries, we have to look at something called the ovarian cycle, okay? And the ovarian cycle looks at the development of the ovum. It also looks at the development of that follicle that we see as well, okay? In fact, right here, you can see kind of a timeline of the development of that follicle. Remember, we have these ovaries full of these primary follicles from what happened before birth, and then we see the timeline of them, right? Growing, eventually ovulation occurs, and then we see their degeneration into the corpus luteum, and eventually what's called a corpus albicans. Okay, that means white body. Corpus luteum means yellow body. Right? And this phase, where we see that primary follicle going through all of this, is approximately 28 days long, with ovulation occurring approximately on day 14. Okay. So that's all fine and good, but we also have to look at what's happening in the uterus as well. Remember, if pregnancy occurs, the uterus is what's going to support that pregnancy. And we have to have the uterus ready for implantation to occur if fertilization occurs. So we also have the uterine or the menstrual cycle that we're also paying attention to. Okay, and that's what we see down here in the bottom frame in this diagram, is we see the uterine or the menstrual cycle occurring as well. So what we're looking at here in this diagram is we're looking at the thickness of the uterine lining, called the endometrium. Okay, so right here at the very beginning of it, you'll notice that it's not very thick. All right. The assumption here is that pregnancy did not occur in the previous cycle. And as a result of that, we start the cycle with menstruation, with the menstrual phase of this cycle. We're sloughing off that endometrial lining during menstruation. All of that epithelial tissue, those blood vessels that grew in there get sloughed off. And then we start to regrow a new endometrial lining in the latter part of the cycle. But the cycle begins with menstruation. We see the proliferation of the endometrium getting thicker and thicker and thicker. This is going to allow implantation to occur. And then if pregnancy does not occur, we see that it becomes sloughed off again towards the end, and we start over in the next month. Okay. So how do we stop this? Okay, how do we stop this from occurring? If pregnancy does occur, if we have implantation of an embryo here into the endometrium, how do we prevent this from getting sloughed off and this from occurring? Well, there's a lot of hormones at play. One of the most important hormones at play, though, is a hormone called progesterone. We talked about this when we talked about the endocrine system earlier in the class. Progesterone, progest, in favor of gestation. This literally means in favor of gestation. Look at what happens to the levels of this hormone as we look at this 28-day cycle. You'll notice that the levels are very, very low here. And then right at day 14, you'll notice that the levels start to go up considerably. Okay. At day 14, that's when we have ovulation occurring, and that's when we have the development of the corpus luteum. The corpus luteum, this degenerating follicle, starts to produce massive amounts of progesterone. Okay, and what that's going to do is it's going to prevent menstruation from occurring. Okay, So the levels go up and up and up. And then if pregnancy hasn't occurred, that corpus luteum's eventually going to degenerate and the levels of progesterone will come back down. That will trigger menstruation to occur. Right? If pregnancy has occurred, by the time this thing degenerates and these levels of progesterone gradually drop off, that by that point, the placenta will be making its own progesterone and the levels will stay high for the duration of pregnancy. All right, so the corpus luteum sort of starts the process, and then if a placenta forms because of pregnancy, that will sort of finish the pregnancy off for the next 9 months. The only other thing worth noting on here that I really want you to pay attention to is look at what happens. Ovulation is here on day 14. There's a really big spike in the gonadotropins, FSH and LH. Those are actually going to be the trigger for ovulation to occur. All right. Okay, onward to the next thing that we have here. I want to talk a little bit about procreation, the act of coitus, which is potentially the most unsexy word in the English language. But the act of coitus is the act of sex. All right. It's actually divided into 4 phases. The first phase is what we call the excitement phase. We then have the plateau, orgasm, and resolution. So what do these 4 phases look like in each of our sexes? In males, excitement is really just the erection of the penis. Okay, I think I have a slide in here as well about erections. I do. So erections are due to parasympathetic vasodilation of the penis. The blood vessels get very large. Okay, and they essentially flood the penis with blood. Okay. In addition to that, though, what they do is because they do flood the penis with so much blood, what happens is the blood vessels that would normally drain the penis of blood actually get pinched. And because of that, we have blood coming in, but it can't leave. And it stays there, and we see erection occurring. Okay. So blood accumulates in the penis relatively quickly. In females, again, because we do see these kind of homologous structures, we see the clitoris becoming more sensitive and erect. The vagina will actually lubricate itself in anticipation of receiving the penis during intercourse. The vagina also elongates just a little bit as well in order to accommodate the penis. All right. So we see that happening here as well. It's not that long. Don't think too much of yourself, guys. It's not getting that big. It's just a little bit. All right. But at any rate, that's essentially what we have here is the vaginal lubrication. The plateau phase is much of the same. Okay. There's really not a whole lot of other stuff that sort of occurs there that is really super noteworthy. Eventually, orgasm occurs. Hopefully, we see that in both sexes. We see the series of muscular contractions, pleasurable sensations, increased blood pressure, heart rate, respiratory rate. In males, orgasm is almost always accompanied by ejaculation, but not always. And in females, we see a contraction of the uterus as well as the vaginal walls. You can imagine that the whole idea there is that we're trying to bring semen in. Okay. Emission, an erection here, we see or I guess we already talked about erection. Emission is where we see the movement of sperm out of the vas deferens into the urethra, where the secretions from those accessory glands, the seminal vesicles and the prostate gland can combine to make semen. The average ejaculate volume in humans is about 3 milliliters. Even though it's less than 10% sperm, we're still looking at like 2 million sperm per ejaculate. So a significant amount, okay? It's worth mentioning that both erection and ejaculation can occur in the absence of any mechanical stimulation. One of the most noteworthy examples of this is the non-sexual erection and emission that occurs with REM sleep, right? Mostly during puberty. I think the colloquial term for those are wet dreams, right? That's what's kind of happening there. If fertilization occurs, okay, it occurs, as I mentioned, fairly far up in the fallopian tube. Okay, the sperm are going to swim from down here all the way through the uterus, all the way up the fallopian tube. Most of them are going to die, I guess you could say, along the way. The vaginal and the uterine environment is fairly acidic. We can only buffer them so much, and they just aren't going to make it. So of the 2 million or so sperm that are ejaculated, only about 40 or 50 actually make it to the egg. It's a really remarkable process. And that's a huge difference in numbers right there, by the way. But as I said, fertilization typically occurs fairly far up in the fallopian tube. As soon as fertilization occurs, there's a series of chemical reactions that will prevent any other sperm from fertilizing that egg, and then mitosis begins immediately. We see that single cell dividing into 2, those 2 dividing into 4, and so on and so forth. Not only are they dividing, they're also starting to migrate, and they're going to eventually create the cell layer that's going to become the placenta. That's this trophoblast, and then that little inner cell mass is going to become. Okay. As I said, prolactin levels remain high throughout pregnancy. You can see here that they rise and rise and rise and stay very high throughout pregnancy. Another hormone that is produced is a hormone called HCG, or human chorionic gonadotropin. This is produced by the forming placenta. And this is actually the first detectable hormone of pregnancy. And we're looking for the presence of HCG in pregnancy tests, because that would be the first indicator that pregnancy has occurred. All right. You'll notice that prolactin. I made a mistake. I was ahead of myself here. Progesterone levels stay high throughout. We see an early spike from the corpus luteum, and then as the placenta develops, progesterone levels stay very, very high for the duration of pregnancy. As pregnancy progresses, thank you for correcting yourself, Dr. Russell. As pregnancy progresses, prolactin levels start to rise. This is going to prepare the breast to produce milk once we give birth. And then we also see one other hormone that helps the breast to prepare themselves. It's called human placental lactogen. It doesn't come online until about the last third of pregnancy, but the levels are very, very high. All right. Finally, we have childbirth. Okay, childbirth or parturition occurs in 3 phases. There's the dilation of the cervix. That's that positive feedback loop that I talked to you about earlier in the semester. As the cervix gets bigger and bigger and bigger to allow for the passage of the baby. There's the delivery of the infant, and then finally the delivery of the placenta as it detaches from the uterine wall. And then you're gonna find yourself that years have passed, and you're spending thousands of dollars to put braces on that kid. Parenthood, gotta love it. Anyway, that's a very quick tour of the reproductive system. So anyway, congratulations on completing the class. Hope you learned something that you didn't know already, and we'll talk to you guys soon. Bye-bye.